This right here is why Mallory's is considered the gold standard. Just look at these 3 examples. Over on the left, we have fibrosis assessment. You can see those brilliant blue collagen fibers just weaving around the pink tissue. That visual is how a pathologist can literally quantify scarring in an organ. Now, look at the middle image, tumor stroma evaluation. See how the blue connective tissue forms a kind of scaffold around the pink tumor cells? That gives us crucial information about how a tumor is structured and how it might behave. And finally, on the right, for connective tissue disorders, the stain highlights these thick, wavy blue bands of abnormal collagen, a dead giveaway for certain diseases. So how does it actually pull this off? Well, it all comes down to what's called the polychrome principle. You can think of it as a friendly competition for the tissue. As the diagram shows, you've got multiple dyes in one solution. The collagen fibers, which are negatively charged, have a strong attraction, a selective affinity, for the positively charged aniline blue. At the very same time, the muscle tissue is attracting a different dye, acid fuchsin. The end result is this immediate, beautiful visual discrimination. As you can see in the key, collagen turns blue, muscle and nuclei pop in red and purple, and fibrin shows up as orange. It's an elegant and incredibly effective system. You know, a perfect stain doesn't start with the dye, it starts with the prep. These pre-analytical variables are absolutely critical. Get any of these wrong, and your results will suffer. First, fixation. You need to ensure your 10% neutral buffered formalin has fully penetrated the tissue to lock in its structure. Second, processing. If you over-dehydrate the tissue, the fibers can shrink, which will destroy the whole picture. Third, section thickness. You're aiming for a sweet spot of 4 to 6 micrometers. Any deviation, and you lose that crispness. Fourth is slide adhesion, and this one is a huge deal. You have to use coated slides, because if that tissue section floats off halfway through, it's game over. And finally, contamination control. Even a tiny bit of water residue or dust can throw off the color intensity. Meticulous is the name of the game here. And here we have it, the entire 6-step staining workflow. Let's walk through it. Step 1 is prep. This is where we deparaffinate and rehydrate the tissue, taking it from xylene through alcohols and down to water so it's ready for our aqueous stains. Step 2 is mordanting. We treat the slide with phosphomalabdic acid. Now, a mordant is like a chemical matchmaker. It helps the dye bind more strongly and specifically to the tissue. In this case, really helping that aniline blue grab onto the collagen. Step 3 is the main event, staining. The slide sits in the Mallory's polychrome solution for 15 to 30 minutes, allowing all those different dyes to find their targets. Step 4 is a quick rinse in distilled water to get rid of any excess dye that hasn't bound. Step 5, differentiation, is a fine-tuning step. A quick dip in 1% acetic acid sharpens the color contrast. And finally, step 6 is finishing. We dehydrate the tissue all over again, clear it, and mount it to create a permanent record.
Let's zoom right in on the molecular level with this fantastic analogy, the ionic handshake. It really captures what's going on. On one side, you have the tissue, the collagen fibers, which are anionic, meaning they're negatively charged. On the other side, you have the dye, aniline blue, which is cationic or positively charged. The mechanism is that simple, powerful attraction between opposite charges. It's a literal handshake between the molecules of the dye and the tissue. And what's amazing is that this is happening simultaneously with all the other dyes and their respective tissue targets. It's this beautiful molecular dance that gives us that stunning final result.
Now this is what you want to see under the microscope. Let's take a little tour of the microscopic appearance. The first thing that really pops is number 1, the collagen fibers, in that deep, vibrant blue. Contrasting with that is number 2, the muscle fibers, which stain red. You can see the nuclei at number 3, which are a deep red or purple. Number 4 is pointing to fibrin, which picks up an orange-red hue. And notice number 5, the elastic fibers. They stay pale pink or yellow because they don't really interact with these dyes. As the diagnostic note at the bottom says, this stark contrast allows for immediate identification of fibrosis. That dense blue collagen just leaps out at you against the red muscle. It's unmistakable. But what about when it doesn't look so perfect? Let's talk troubleshooting. If your collagen looks pale and washed out, the cause is likely your reagents. Maybe your aniline blue solution is old, or the Morton step was too short. The solution? Prepare fresh reagents. If the muscle is way too red, overpowering everything else, the cause is usually overstaining. You've got to shorten your time in the polychrome solution. If you've got too much background staining making things muddy, the cause is insufficient differentiation. The solution there is to increase your time in the acetic acid. And finally, if the tissue is literally lifting off the glass,
That's a slide adhesion problem, pure and simple. You need to go back and ensure you're using the right kind of coated slides.
So we've covered the science and the hands-on procedure, but none of that is worth much if it isn't done within a framework of excellence. That brings us to Section 4, Quality, Compliance, and Safety. This is all about the professional standards that ensure our results are trustworthy and that we're keeping everyone in the lab safe.
When we talk about ISO 1589 compliance, it's not a one-and-done thing. It's a cycle of continuous quality improvement. It all starts with validation, where you rigorously prove that your method works in your lab. Then comes daily internal QC, running control slides to make sure everything is performing as expected. All of this has to be backed up by meticulous documentation. If you didn't write it down, it didn't happen. And of course, there's competency, ensuring the staff are trained and proficient. The whole point of this entire system is risk management. It's about catching potential problems before they can ever impact a patient's diagnosis.
Okay, I want to pause here because this could not be more important. Let's talk about safety and material handling. The chemicals we use are serious. First, aniline dyes. You see that skull and crossbones? That means they're toxic and potentially carcinogenic. Next, the acids, like acid fuchsin. That symbol means they're corrosive. They can cause severe chemical burns. Then you have the solvents, like orange G. The flame means they're flammable, but they're also neurotoxic. They must be used in a fume hood. The rules are simple and absolute. Personal protective equipment, your gloves, lab coat, and eye protection is mandatory. And all waste has to be handled according to strict environmental and safety regulations. There are no shortcuts with safety. Period.
All right, we have covered a ton of ground, from deep dive chemistry to lab safety protocols. So for our final section, let's bring it all together and consolidate our knowledge with the summary and key takeaways. If you only take away 3 things from this entire explainer, let them be these. First, the principle. It all boils down to the simple chemistry of ionic attraction. Opposite charges attract, creating distinct colors. Second, the compliance. Reproducible diagnostic results are only possible through rigorous quality control and standardization. And that leads us to the third and most important point, the golden rule. Standardization is everything. It is the absolute key to accuracy. And when you follow that rule, you get the beautiful result you see on the right: blue collagen, red muscle, purple nuclei, and orange fibrin.